so hard. Bum, 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 bum. Yep. Why? আমি এখানে কথা বলতেছি, আমি কথা বলতেছি। What do you mean কেন আমি কথা বলতেছি? No. Still, আম্মু, please. আমি পড়া করতেছি।
Is he studying? Yes. Okay. আমি নাও ঘরে নামাজ পড়তে যাচ্ছি, তুই আমাকে রিপোর্ট দিবি। আর তুই আবার কথা বলতে লাগিস না ওর সঙ্গে।
ওই আসলে বেশি কথা বলতেছে, ওকে একটু চুপ থাকতে পারে। Okay,
আম্মু প্লিজ।
place.
এই টেবিল আমার দিকে সরাইতে হবে।
I don't want this to show my whole screen because that is kinda mean. And I am poopy pee. What the hell? Please. No, it doesn't show. Why? Why do you not show? The answer is simple. I just need to go here, move my thing here. Anything else that I want to move there? Not really, other than this. And yeah, we good. We bing chillin' flingin'. Throw in ChatGPT over there. Throw in some Gemini. Throw in some Gemini. Get this in here. We're gonna make a document. What the hell? I mean, yeah, the UI in this app is pretty nice. I'm not gonna lie. Blank note. We're gonna move this down to the end of chapter test. Oh crap! I didn't do the statistical tests. That's true. Okay, it's 2 pages of pure poopy. I've been staring at you for the past like 10 minutes, hoping that you would look back. I'm just like, I wanted you to just turn back on me and just get scared because I've just been looking like this at you. Man, why so stupid?
That's why it's not usually quiet.
Yeah, I wanted to be like, hey, what's going on? Why is my paper is like this? Oh hell no! What the hell happened? What is this space over here? Oh, there's an issue. There's an issue that I can't solve. You ain't solving nothing.
Allow me to lock in.
Allow me to flop in. Flopping. I have to do this on real paper, not on that digital paper. The big boy paper. Sagaru, which means to raise, and it's gonna be intransitive. It's too easy. Geico. Diplomacy.
Transitive and intransitive
bloody ante.
Because what's the basic economic problem?
It's like the supply demand keeps increasing, but supply doesn't.
Yeah, the scarcity of resources. Demand for resources is greater than their supply. We just agree at hell, bro.
And what am I doing? Okay. Yeah, I have to do this on paper. I have to do this on paper. Okay, here we go. So therefore, this needs to go. I need my fountain pen, my mighty fountain pen that I love so dear. I love you. Yeah, my mighty fountain pen. I love you so much. Oh,
You shut the hell up. What the hell are you doing?
yeah. What?
Dude, honestly, I was reading it and then I heard what you were doing. Dude, I was about to say some very bad words, so like, I had to stop myself.
What? Why? I just love a fountain pen, bro.
Shut up.
Bruh. Can't have nothing in this family. Oh my god. Okay, yeah, maybe I went too far on that joke.
I was about to say, like, I felt bad words on the tip of my tongue. I don't know where they came from. I don't know where.
Yeah, I knew you always hated this fountain pen. I knew. It's okay. It's don't cry, don't cry. I will always love you forever, fountain pen. It's okay.
You solved your exam?
Just kissing my fountain pen during the exam.
Exam hall, all people do it, just the kids and everything. You can't do it.
Teachers like just want you. That used to be me when I was 15. Brings back memories. Okay. Okay, lock in. Fatima, lock in. Faisal, lock in. Alright, question 1. The following substances are all involved in photosynthesis. 1, 2, 3, 4. Glyceraldehyde, phosphate. What? Is that what GALP is? GALP? Oh shit. Okay. Glycerate 3-phosphate, I'm guessing that's G3P or GP. Carbon dioxide and glucose. Which order of substances will the carbon atom be transferred? It's 3, 2, 1, 4. Question 2 is, what are the products of light? What are the products of light independent stage of photosynthesis? Light dependent, sorry. That's going to be ATP and reduced NADP. So that's going to be D. How many molecules of ribulose biphosphate are broken down to produce 2 molecules of glucose? Okay. This is the annoying part. Okay, so 2 molecules of glucose means glucose is a hexose sugar. Therefore, you need 12 carbon atoms, and you know that you only have what's it? It's 6 every 2 gallops. No. You get 2 gallops every 10 gallops. Or is it 2 gallops every 12? Yeah, it's 2 gallops every 12. 2 gallops means 2 times 3, which is 6. So therefore, if you need 6, man, this is a hard question. What you talking about? Okay, first of all, let's see. You need 12 carbons. Therefore, you need 4 gallops. Therefore, you need 2 and 2. And you know, for each 2 gallops, you need 12 gallops. And for 12 gallops, you need 12 RUBPs. So therefore, it's 24. No. Ribulose biphosphate is RUBP. How many molecules of RUBP is broken down to produce 2 molecules of glucose? Okay, no, sorry. Okay, think about one glucose, which is 2 gallops. You need, it's 2 every 12. And 12 is 6 RUBP because it's 3GP and 2 gallop. So it's 6 RUBP. And so if you're looking for 4 gallop, it's 24 things, and so that's 12 RUBP. So it should be B, I believe. Okay, finally comes to the writing. Okay, so it's saying that one of the reactions of photosynthesis can be summarized by shown below. Water to hydrogen ions plus oxygen gas plus electrons. Name the reaction. That's going to be photolysis. State one other factor not shown above that is required for this reaction to occur in a chloroplast. That's sunlight, right? Describe the roles of the electrons in the light dependent stage of photosynthesis. Just want to quickly just check. Okay, I've just been displaying nothing this entire time. Um, yeah, I've literally just been showing nothing. That's a bit funny. Okay, it's fine. I haven't been. I mean, I haven't been writing anything on the screen, so it's okay. Okay, so what does the electrons do? The role of the electrons. I feel like the main role of the electrons is to be passed around the protein carriers during the electron transport chain to make the ATP, as well as fill the hole that's left in the PS1. Maybe I wouldn't really say that because those electrons are emitted like once again, but they are definitely used to reduce NADP to NADPH. Anything else? So electrons used to produce ATP by being carried around through the ETC, also used to reduce NADP to NADPH. It is used to fill back the hole in the cyclic photophosphorylation. Can I even say that? That is not right. Okay, so I'll just say so to make the reactants used in the light independent stage, light independent LIR, light independent reactions of photo no, no. Oh my god, I hate this part. It's okay. It's okay.
Why are you being so aggressive?
Please. No. Breathe. Breathe. Try. Breathe. Shit. Why isn't it doing?
Don't you have to wait a little bit?
Yeah, but okay. We're good. You don't necessarily have to. If you don't, it just comes a bit thicker. And since I'm already writing, I don't want to wait. Okay. Essentially, I'm torturing the pen. Barnabas. Sorry. Explain how the products of light-dependent reaction are involved in the products of GALP. So, That's 5 marks? Are you crazy? The hell? Okay, here's 1 mark, here's 2 marks, maybe 3rd mark? What is the 4th mark? It's describe. This is explain. So first let's describe and then explain. So or and then give the reason. So first I'll say n sorry, reduced NADP is used to donate its hydrogen to GP so that it can have it so that it can have does GP is no, yeah, the aldehyde group requires the H, right? An aldehyde group ATP used to power the reaction to convert GP into GALP. Explain how the products are involved in the production of this. So reduced NADP donates hydrogen to GP so that it can have an aldehyde group. ATP used to power the reaction to convert GP into GALP. Those are the only 2 products. Without these 2 products, without these 2 products, GALP cannot be made. I don't really know what else to write, so I'll just keep going. GALP does not accumulate in chloroplast during photosynthesis. State how GALP is used following its production. Used to make, used to regenerate RUBP, make hexose sugars like glucose and proteins and nucleic acids. Okay, question 5 now says diagram summarizes light dependent reactions. Light, chlorophyll, electrons to electron carriers to product A, electrons to product B. What? What shitty thing is this? Is this talking about? Okay, maybe I kind of get what they're talking about. Why are we doing lasso select? Let's just. Yeah, so far it's okay. No, it's not too bad so far. It's not that bad. It's okay. Why are you not panning? Do you only pan with the stylus? Okay, I have a feeling it's something wrong with this. Was it this guy? Is it this? It's your fault, isn't it? You did something on my laptop while I was gone. Yeah, I knew it. What did you do? What did you do? Don't lie to me. You are H. Come here. Come here.
Ow! Stop! Ow! Dude!
Yeah. Yeah. No one can save you now.
Ow! Stop!
Boom. Come here. Oh. What you said about fountain pen? Just a prank, guys. Just. Why are you resisting? It's just a prank. Back to task. Wait, wish you actually die. No. Such a bad idea. It's just this guy was doing all of that. Oh, we have 10 viewers. Oh no.
Actually getting cancelled in biology.
Okay. State the precise location within a chloroplast where this sequence of reactions occurs. This is going to be happening in your thylakoid membrane. Your thylakoid membrane. Yeah. You solve your own hunger. We're good at kitchen. Okay. Let's see. This needs to be in the thylakoid. For the way you need to. Sorry, I'm gonna stop talking. Product of name of product A. That's going to be ATP. Name of product B, that's going to be reduced NADP. And H, it's going to be option D and option C. Okay, then they ask, explain why the chlorophyll in photosystem 2 loses an electron. Because so what am I supposed to say? Chlorophyll, no, when a photon with the right wavelength hits the P680 chlorophyll protein, it absorbs the energy. Well, it more like it traps the energy, but it absorbs the energy from the light and uses it to excite an electron the excited electron leaves the chlorophyll and gets and gets binded to a protein carrier, or is it electron carrier? I don't know. Okay, D says chemical called atrazine prevents the flow of electrons. It's always questions like this. Let's just got that. For me, you need to get a fountain pen, bro. I'm telling you, the writing experience is so different. You're gonna have this one, but if I have this, if I give you this one, then I'm gonna get a really nice one next, because I want to invest my life into fountain pens. I'm not even joking. Like, fountain pens are so nice. Anyways,
Basic economic.
okay, chemical atrazine prevents the flow of electrons to electron carriers. It's called an electron carrier. Okay. I hate when people use the phrase "knock on wood," like, and they actually do it. I'm gonna slap your face like so hard right now.
Can you shut up and study? Like, what are you talking about?
Like, literally, I'm gonna knock on your face. What?
Knock on wood. I don't know what it means, by the way.
It's like this. I don't know, man. It's like, oh, don't call the devil, and it's going to jinx you only. Oh.
I'm going to tell them you're not studying.
What do you mean I'm not studying? I just said something. Because I like saying something. Got a problem? Bloody onto it, eh? Describe the likely effect of atrazine on the production of carbohydrate in a chloroplast. So, the effect of atrazine on the production of carbohydrate in chloroplasts. So, atrazine causes, okay, I need this to be on top. Causes, I think it's better when I do this, but yeah. Causes what? What does it cause? Causes less ATP to be made. It also causes what does it cause? Less reduced NADP to be made. As a result, the light independent reactions are slowed down and less Gaup is made, thus it's not less Gaup is made. If atrazine prevents the flow of electrons, therefore it's no ATP to be made. It also causes no reduced NADP to be made. As a result, LR reactions are stopped, and no Gaup is formed. Thus, no glucose or carbohydrates can be made. Okay, 6. The diagram shows what happens to electrons during a part of the light-dependent stage of photosynthesis. Any excited electrons that are not taken up by electron carriers follow pathway A and release energy as light in a process called fluorescence. I see. The excited electrons that are taken up by electrons follow pathway B. So you have X, which I'm guessing is chlorophyll, and so the electrons go to the electron carriers, and if they can't be taken to the electron carriers, then they go back to the chlorophyll, but release the energy in the form of light, and then the ones which do get binded to the electron carriers move to B, where they go through the electron transfer chain. What is Y? Where they bind with NADPH to make reduced NADP to make reduced NADP. So X and Y. So X is obviously chlorophyll, and Y is your boy NADP. Dude, you should be using my textbook. I have like a lot of like highlights and notes on it. I think I made some annotations, but my annotations are unreadable, so about that, I guess. But
We have it. Maybe I will.
you should try looking at it, maybe. Man, I hope you have as much fun as I did. Cause man, Sonali made those classes so much fun. Man, those are such fun times.
Yeah,
You're gonna be asked to make a lot of PPTs. You're gonna make, you are going to be made to. You're gonna have to write a lot, and you're gonna kind of hate it because the exams are very, 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 very structured, and it's almost inhumane how structured it is. Because you don't write like a normal human. You write the way the exam papers want you to, and they structure the learning around that. So,
why is the school system so terrible?
because the British.
But I mean, I feel like the American system isn't as great either.
Honestly, I don't know. People say that, but I mean, the greatest.
I keep on hearing like college students are always like the moment they're done college, they're in like serious debt now.
Oh, I mean, yeah, because there's a very big problem with like school debt. I mean, like, yeah, school debt. Like how like colleges are super expensive, so like, basically it's just student debt. Like they don't the parents can't afford university, but what colleges allow is like the student can study, and even if they can't afford it, it's fine. It's just the student has to pay the student debt after they graduate, and sometimes that student debt is so large that like the first like 10 years of them getting a job and earning goes straight to their student debt. It's that bad.
Yeah.
But I mean, that's how wealthy we are that we don't need to do that because Algo can afford all of it. Yep. Anyways, explain the importance of reduced Y in the process of photosynthesis. How many times, brother? Okay. Reduced NADP can donate its hydrogen to GP to form GALP, which is needed to form glucose. Reduced NAD can donate its hydrogen to GP I think I should just mention in the light independent reactions. Okay. Then C says a light was shown on a leaf and left switched. Huh? A light was shown on a leaf and left switched on. Okay. The graph shows changes in the amount of light given off as fluorescence by the leaf. So time since light was switched on, fluorescence. It increased and after 1 second it decreased. And then it stopped entirely. So just the explanation for the increase in fluorescence. Is it like, so the light is getting like, so the light turns on and so it starts really dark and it gets brighter and brighter and brighter. And so, no, because they get excited and then they stop. What the hell? So what's going on here? Electrons are being excited, they go, what is it? They are not taken up by electron carriers. Why not? So the increase in fluorescence is that explanation, right? So as light is turned on, more so the decrease in fluorescence could be, this is all C? Yeah. This would be maybe Explain why an inhibitor of carbon dioxide fixation will lead to an increase in fluorescence. If carbon dioxide got fixated, by something else, so then you would have no GP being made, which means that your electrons okay, so think about the end result, which is Y, right? If Y is not present, therefore it goes back to A. So if, okay, talk about D. If CO2 gets inhibited, NADPH does not get oxidized back to NADP. Thus, excited electrons cannot get accepted by electron carriers and follow path B. So it is path B as Y is not present. Thus, it is forced to go through path and release fluorescence. Seven. A. A is a thylakoid and B is a granum. So that's going to be P. B says, state precisely where chlorophyll is found. It is found on the surface of thylakoid sacs in the chloroplast. C says, a student investigated. Let's turn this to the next page. A student investigated the. I hate these questions, man. Okay. The The student investigated the effect of different colored, let's just have this here. So the effect of different colored lights on the rate of photosynthesis. What is going on with this app, bruh? Why, what is this? Like, what are we doing here? What are we doing here, man? Should I restart? I think I should restart. Okay. In that case, so the app is working. Nice. And that is the exact moment I'm restarting. Ain't that nice? Okay. So we're gonna be restarting this. Okay, now it works. Yo, this is... I'm confused. Okay, whatever. Let's just fix this. I know I shouldn't be using... Oh, it's a bit big. Oh. Can we just fit it, please? Let's see. Transform, fit to screen. Yet it's a bit too big. I'll tell you what, that's okay. You know, Pun doesn't care. Okay, so we have so different, and it's broken again. What are we doing here, app? What are we doing here? Okay, the effect of different colored. Wow, I'm bad at this. Different colored lights on the rate of photosynthesis. She placed. Okay, Bada. Sorry, my apologies for trying to do this. Different colored lights on the rate of photosynthesis. She placed. What'd she do? She placed your boy. She placed colored filters on the lamp. Between the lamp and a beaker containing a piece of pondweed, counted the number of bubbles released from the pondweed in 2 minutes. Okay, different colors of light means different wavelengths of light. Different wavelengths of light means some get accepted, or more get absorbed, less get absorbed. As more wavelengths of light get absorbed, therefore more electrons are emitted, which means more photosynthesis occurs. So state the color that, okay, so it was red. 2 asks, explain the results shown for the black filter. So, okay, the light that passes through the black filter contains wavelengths of light of which most are absorbed. It's 2 marks. I am about like
Beginning.
no. Last question is question 8. I'm on question like 7 on the last few parts.
Okay, she's good.
Can you please close the door? As a result, Now are electrons emitted more phot- green is bubbles less. It's not more photosynthesis occurs, it's more photolysis occurs. Less. Okay, so light passes through green filter. Wavelengths of light are not absorbed by chlorophyll IV says the student expected to be this is I wrote too much for 2 marks. The student expected that there to be no oxygen released when using the black filter, so just 2 reasons why some bubbles were observed when the black filter was placed. Oh my god, I thought it said blue. That makes it a lot harder. That makes it a lot harder. How do filters work again? Okay. Okay. Explain the result, right? So explain why it's the least and explain why green is a little bit higher, okay? So okay. The wavelength of the light that passes. No. Okay, 3 can be explained by saying green filter allows more wavelengths of light to pass through it compared to black, so it
Spanish restaurant color. Restaurant rice and soup. Spanish restaurant.
way Peter Han did it was a lot better than me now. Anyways, what are we doing? What are we doing here? The second reason was perhaps, so this is what I was thinking. Still performing photosynthesis using light from using energy or using trapped light from previous test. So there's two ways in which this could have been improved. Perform experiment in a dark room. Wait a longer period of time. Between tests, or use a dormant, or like use different on-bead sample for each test. Okay, last question. So here we go. The carbohydrates in green plants are formed during the light independent stage. They are synthesized from GP. Say precisely where the synthesis of carbohydrates take place. The stroma. This is A. B, I says name the products during the light dependent stage that is used during the synthesis of carbohydrates. So that's going to be reduced NADP plus ATP. Name the source of carbon used to manufacture sugars. The source of carbon, CO2. C says, what is the role of RUBP? Is it to convert carbon dioxide to GP? It is used to fix carbon dioxide, I would say. Accept electrons, release energy. It is used for carbon fixing now. Like it binds with carbon dioxide with the help of Rubisco. So I would say it's B. Okay, we have already seen this graph, so maybe we can make this a bit easier for ourselves. আমি কি পেপারটা আজকে এখনই মার্ক করবো নাকি An investigation of photosynthesis in cells taken from green alga was carried out. Samples of algal cells was taken at 1 minute intervals over a period of 6 minutes. How long have I been studying for? Just curious. It's like 40 minutes, right? An hour. Damn. Sorry? Oh, I'm not sure. Let's see, when did I start? I started about Yeah, I started an hour ago. Maybe I took 10 minutes in the middle away from studying. So that's 50 minutes. 50 minutes for this. How many marks are there in total? 9, 12. So that's 21. 21 and then 13. 13 plus 21 is 33. 33, 43, 43, 1, 2, 3, 46. So I'm still not done actually. So let's say this takes like another like 15 minutes. So maybe I took a bit more time than I should have. Yeah, I did make a few mistakes. So yeah, I did take a little bit extra time. But not too much, like a dangerous amount. Like it's just a bit of extra time. I could have probably, if I worked harder, maybe I could have saved like 20 minutes or a bit less than that. Okay, anyways. Samples of algal cells were taken at 1 minute intervals over a period of 6 minutes. The quantities of GP and RUBP in these samples were measured. At the start of the investigation, the algal cells were kept in an atmosphere with 1% carbon dioxide. After 3 minutes, then it was moved, the carbon dioxide concentration was decreased, and here we have the thing. So describe the effects on the quantities of GP and RUBP with the decrease in carbon dioxide and deduce the explanations for the effects you have described. So 2 effects and 2 explanations, I'm guessing, because it's 4 marks. So first, just describe what's going on. So effects on the quantities of GP and RUBP. So at 3%, at 1% carbon dioxide, RUBP and GP are at constant quantities and there is more GP than RUBP, since why? Since is it because there's 2 GPs made for every RUBP? Right? Since 2 moles of GP is made per every mole of RUBP consumed. After 3 minutes, CO2 concentration is reduced. RUBP quantity is increases, and then decreases. GP quantity decreases, until a new equilibrium is reached. More RUBP present. More RUBP than GP present, since carbon fixing slows down and RUBP is built up. That's my answer. So it's about 3 pages of answers. Okay. Holy crap, what the? I don't know. Anyways, my answers are somewhere, so let's just bring them up. Open from device. Study materials, bio. It's gonna be SB2 answers. SB2 answers for me and for you. What are we gonna do?
How's it?
Oh, I didn't mark it yet. I have not marked it yet. 5A. Okay, so I'm supposed to mark this. I'm not very good at marking, but I'll try. So CDB, That's 1, 1, 1. Photolysis. Sufficient light intensity. I read sunlight. That's the same thing, I hope. Okay. Electrons combine with H plus and NADP to produce reduced NADP. Electrons, okay, and then electrons absorb energy from photons. Electrons carry the energy into electron transport chain. Electrons pass from carrier to carrier in the electron transport chain, releasing energy in small amounts. I wrote electrons used to produce ATP by being carried around through the electron transport chain. Pass from carrier to carrier in the electron transport chain, releasing small amounts of energy. I wrote produce ATP. Produce is the wrong word for sure. It's release. You don't produce energy, you release energy. So there you have one. Electrons carry the energy from the photons into the electron transport chain. And electrons absorb energy from photons to become excited. It is used to reduce NADP to NADPH. Electrons combine with H plus and NADP to produce, okay. Electrons don't reduce NADP to NADPH. Yeah, that's wrong. This is unnecessary. And this is wrong. Electrons used to produce, okay, so this has to be release ATP by being carried around through the ETC. Being carried around is not a good thing. It should be by being passed from carrier to carrier. Passed from carrier to carrier. Okay? This is used to combine with NADP plus H plus ions to form NADPH. Okay? Okay, so how many marks am I getting for this? Oh, God, I have to do this every single time. Okay. That's going to be... so that's 1 mark. That's 1 mark. Produced to release is, I think that's fine. By being carried around to the ETC, by being passed on from carrier to carrier, it's honestly the same thing. So I will be describe the role. Oh, there's 4. Oh crap. Okay, I didn't do. Okay, so absorbing energy from photons is actually one of the. Damn. Okay. Carrying the energy into the electron transport chain is actually one of the roles of ATP. Interesting. Okay. Sure, you can consider it that, I suppose. That's actually an interesting way to say it, so I'll write that down. Role of ATP is to actually absorb the energy from incident photon and carry it into the ETC. Okay? The role of ATP, bro. The role of electron. So I'm just going to say this is 0. Then reduced NADP is used to donate. Okay, what's the next question? It says, explain how the products of LDR is used in glyceraldehyde production, GAL production. So transfer energy from transfer energy from LD stage to LI stage. Energy is used to make bonds, converting GP to GALP. GALP to RUBP? That's right. Yeah, that's also true. Yeah, forgot about that. Transfer hydrogen atoms from that to that. Hydrogen atoms you use to reduce a GP to GALP. Okay. So wording needs to be different for sure. Yeah. It's always wording problems. That's A level for you. Oh my god, I bet that guy is gonna make such a big deal out of it. It's gonna be so much fun. Okay. So reduced NADP donates its hydrogen to GP so that it can have an aldehyde group. ATP used to power the reaction to convert GP into GALP. Okay, so this is the wrong idea. Just pause for a second. Alright, let's continue. So, let's see. Let's open GPT for us. Let me put this on. How do I do this? I have to move this towards this, so I can then put on this. Okay. So then we can turn on PDF write rather than PDF left. PDF write is going to be turning on this. It doesn't work, does it? Okay, it doesn't. What if I save? Will saving fix it? Saving does not fix it. Okay. So let's move them. Oops. Oh, don't tell me. Okay. Okay, that's fine. I'll just keep it like this. So, how's ATP used to make bonds? ATP isn't exactly a raw bond maker. What happens is ATP hydrolyzes ATP to ADP plus PI plus energy. The phosphate group PI is transferred to an intermediate molecule like GP to phosphorylated intermediate. Adding this phosphate is like priming the molecule, making it more reactive. That reactivity allows new bonds to form, e.g. GP to GALP, regenerating RUBP. So when Marx schemes say energy from ATP used to make bonds, the real meaning is ATP phosphorylates molecules, raising their energy levels so bond rearrangements like turning GP into GALP can happen. Really? Hydrolyzing ATP does release free energy. That energy can be coupled to an endergonic energy requiring reaction. This is how it's often described. ATP energy release powers reaction. The more accurate picture is the real trigger is phosphorylation. ATP doesn't just release energy into the air, it usually transfers its terminal phosphate group into a substrate. And when that substrate is phosphorylated, it's in a higher energy state, the less stable state. Because it's unstable, it can more easily undergo the bond rearrangements needed to form products like turning GP into GALP. After the reaction, the phosphate can be removed, leaving a lower energy state, more stable product. So in one sense, yes, ATP is phosphate bound. It does release usable energy, but in practice that energy is transferred by phosphorylating molecules and not just by bond breakage alone. Okay. So that's how ATP, the energy used to make bonds. Example in the Calvin cycle, GP to GALP. ATP hydrolysis adds a phosphate to GP, or to an intermediate step. This makes an intermediate reactive enough to accept electrons plus H from NADPH, reducing it to GALP. Glycolysis is the easiest place to see ATP's real role. Take the first step of glycolysis, glucose to glucose 6-phosphate. So glucose plus ATP makes glucose 6-phosphate, G6P. An enzyme, hexokinase, transfers the terminal phosphate from ATP onto glucose. Now this C6-hydroxyl group of glucose is phosphorylated, giving glucose 6-phosphate. Why do this? The phosphorylation traps glucose inside the cell. G6P's transfer is charged and can't diffuse back out through the glucose transporters. Phosphorylation raises the energy level of the glucose, so now it's in a higher free energy state than plain glucose, which makes later steps like isomerization, cleavage, oxidation easier because they'd be more energetically unfavorable otherwise. So the principle is that ATP doesn't just release energy, but it transfers the phosphate, which makes glucose more reactive, and that phosphorylation couples the exergonic hydrolysis of ATP to the endergonic endergonic reactions of glycolysis. Same thing in Calvin cycle. GP is phosphorylated by ATP. Activation allows NADPH to reduce into GP and GALP. So whenever you see ATP used, imagine it as slapping a phosphate group onto something that destabilizes it, primes it, and makes new bonds possible. How NADPH provides hydrogen to reduce GP to GALP. Here, reduction is literal, gain of electrons and hydrogen. NADPH is carrying 2 high energy electrons plus a proton. It transfers these to GP. This changes GP into GALP. Chemically, GP plus NADPH plus H plus mixes GALP plus NADP plus. No. So NADP donates. Oh, I don't get this. Can I look at some video that can explain it? GP to GALP. to understand. Oh, that's weird. Okay. There was a video. Mm, a lot of info here, man. I remember sending it on Instagram. No, I know. I remember where I sent it. It was. No. It's cause I'm just kind of doing something else. It's related to bio, but it's a bit different. One second. ওর নাম কি ছিল? Rubisco carboxylation. Rubisco carboxylation mechanism. What am I doing? Okay, so carbon fixation, CO2, 3-phosphoglycerate, that's the intermediate, and then G3P in the regeneration. G3P is the same thing as GALF, right? I need to make sure. G3P I need to know the other names. 오케이 한번 회사 아미 어땠지? 뭐 아미 어땠지?
উঠে লাইট অফ করো তারপরে যাচ্ছি
Used to be phosphorylation reduction, right?
So here transformation. So next here you can see the cycle, and this is a Calvin cycle, correct? And in this cycle there are some steps involved, and in that step the main important here is that Ruby scores, or we can say only just tribulose 1,5 biphosphate, okay, the carboxylase oxalate, which will be said as this goes right here, okay. So this Ruby score which is mentioned here, it's a very main important component in the overall this cycle. Basically this enzyme combines the carbon dioxide which is dissolved in the cytosol, okay. As you can see, carbon fixation is done here. So carbon fixation, that means obviously CO2 will involve here, and that carbon fixation in the here you can see CO2 is there, and your carbon fixation is done because of this. Because after that the 3-PGA is formed here, okay, 6 molecules of 3-PGA. So what is this 3-PGA? Basically it is 3-phosphoglycerate, okay, that will be said as 3-PGA. 3-PGA, that is 3-phosphoglycerate, okay, and that 3-phosphoglycerate is here, and this 3-phosphoglycerate will convert into, as you can see, G3P, and that G3P will be said as here this G3P is a conversion, and in this 3GP what will happen to all over reaction? Let's have a look on that. So in this reaction what is the most important thing? Let's see. So here first we will write here acetaldehyde 3-phosphate, okay, and that is for G3, okay. Now let's see what is going to happen here. In this reaction of the conversion, the first thing which going to happen, that first this 3GPA, that will the 3GPA kinase add to phosphate, and here the ATP, the phosphate is taken out from ATP, as you can see this 3GPA, and for that from the ATP the ADP formation is there, as you can see arrow ATP to ADP, because one of the phosphate is being used here, and that phosphate is why it is being used here. That phosphate is being used to make 3-PGA to form a 1,3-biphosphogystrate, okay. So this reaction will be right here. What will happen? So this 3-PGA, it will get converted into 1,3 by here, sorry, that should be 1,3 biphosphogystrate, okay. So for this conversion here, the one phosphate will be used, phosphate, and that phosphate is obtained from, that phosphate is gained from ATP. So ATP phosphate will be used, so that's why ATP will become now ADP. So adenosine triphosphate will become now diphosphate, okay, because phosphate is being used. Now then after that the NADP will be used, or we can say the NADP is removed. As you can see here, the NADPH is getting converted in NADP plus, okay. So here this is using or removing of hydrogen. Why it is taking place? So this removed hydrogen will be used in this 1,3-biphosphogystrate, and what will happen? Let's see. So here you can see the NADPH, and this NADPH, out of this it becomes NADP plus, because the hydrogen pH is being used, and because of this usage of pH, this 1,3-biphosphogystrate, it turns into, or
Okay.